Good morning and welcome to Saipan, everyone. My name is Jill Liskey-Clark. I'm with the CNMI Division of Fish and Wildlife, and I am one of the volunteer organizers of this event. If you need anything at all in the next couple days, come find me. I'll hook you up. Saipan is my home, so I can tell you where to go and what to do and what's going on this week. I'm really pleased to be able to have you all here today. This is the third annual conference, but the first time here on Saipan. We have here today the director of the CNMI Division of Fish and Wildlife, Mr. Manny Pangilinan, and he's just going to say a few words to start us off today.
Thank you, Jill. Afare, good morning in Pirawami. On behalf of Governor Ross Torres, Lieutenant Governor Arthur Palacios, and Secretary Anthony Benavente, I am happy to welcome you to the third annual Marianas Terrestrial Conservation Conference. Like Jill said, this is the first time that Saipan has hosted this conference, and we are happy to do so. And we are excited to share our island with you as well. I encourage those of you that are new to the island to get out this week and see our forest, birds, and other wildlife for your enjoyment. For those of you that are from Saipan, thank you for coming today and showing your support for conservation. I see a fantastic slate of presenters on today's agenda, talking about aga, sanihi, and many other of our endemic species. Thank you to the biologists from near and far that are helping us understand how to manage our islands so people and species can coexist. We appreciate the work you all do. Today's presentations are important for sharing information, but the conservation that occur outside today are just as important. With that in mind, I want to invite you to the poster session and barbecue that will be held tomorrow evening. My colleagues from the Department of Lands and Natural Resources have organized local-style barbecue. I want to share good food, drinks, and conversation. Thank you again for coming. Enjoy your day and the rest of your time today on Saipan. I want to also thank the organizer. I believe that we owe them a round of applause. Thank you, Jill, for making this happen. Audrey and everybody that has volunteered to make this happen. It is not hard work, and I know Jill will testify to that. Thank you.
Thank you, Director. Okay, I am going to pass off the microphone in just a moment to Halder here, who is going to be our moderator today, and thank you to her for jumping in last minute. She looks more frantic than me this morning. Just a couple logistics before we get going. Restrooms, there's the ladies in that corner and men's room in that corner. These are rather small though, so during breaks, if there's a crush, if you just walk out past the restaurants, there's larger restrooms out there. If you need Wi-Fi, the password is at the registration table, but I can tell you what it is right now if you like. When you go in, you have to go in as a restaurant patron, whatever it's, click that you're a restaurant patron, and then the password is Dibuli, D-I-B as in boy, U-L-I. So if you need that. Let's see, grab yourself a pastry. These are provided by Herman's Bakery. Thank you to Herman's for that. And I think that's all we need as far as logistics goes, and I think we are ready to get started. Thanks, everyone.
Okay, good morning, everyone. Thanks for coming out this morning. It's exciting to get this year started, and it was wonderful to have Jill organize this year, and all the folks up here in the CNMI. So thank you for being wonderful hosts and putting on some excellent field trips so far. Everyone I've heard has loved the Swiftlet trip and the Proa rides. So I'm excited for the rest of the day, or the week. I just put up the schedule for today. As Jill mentioned in her emails, we don't have any handouts of it, but you can go to the website and you can download the PDF of the schedule if you'd like. We're starting off with a section on birds, and we have a Zoom call from Henry Pollack, who was with Colorado State University, is now with University of Illinois Urbana-Champaign. And he's going to be talking about ongoing multi-level impacts of the brown tree snake on the behavior, survival, and ecology of Guam's avifauna. He has a Zoom call, so hold on a second, Henry, almost ready. He has a Zoom call, so if you don't need the internet, please don't use it, because the download speed is really, really slow. Hopefully this is going to work, but please let's leave as much bandwidth as possible for Henry. And now I am, one minute. Going to turn it over to Henry.
All right, can everybody hear me?
Sounds good.
Awesome. Oh man, I wish I were with you guys in the warm climes of the Marianas. I'm wearing, I don't know if you can see me, but I'm wearing a scarf right now, so it's a little chilly back here in Illinois, but I'm super excited. This is the second year that I've come in through teleconference, so thanks to everyone, to Jill and Halder for organizing, and I just want to quickly thank all my collaborators. This has been a sort of multi-institutional effort, and is sort of a synthesis of the last couple years of research that I was doing as a postdoc working on Guam's birds. So this talk is going to be, as I mentioned, synthetic. I'm going to skip most of the methods, just kind of present a lot of general results just because I find that more interesting, so hopefully you guys do as well, so I'll get started. So as we all know, Guam is home to the invasive brown tree snake, which was introduced in World War II, sometime after World War II, and had devastating impacts on the avifauna. And Guam was home to not a super diverse, but a pretty interesting avifauna with some endemic species. And in the years following the establishment and spread of the brown tree snake, being modeled by my good friend and collaborator Martin Kastner here, the snake, like I mentioned, had devastating impacts on the birds. So this graph on the right you can see here is from Gary Wiles' paper in the early 2000s. And you can see that around the mid-70s to early 80s, most of Guam's birds exhibited these precipitous population declines. And the brown tree snake ended up causing the extirpation of a lot of Guam's native avifauna, including the extinction of several species from the face of the earth. So now on Guam, we have this sort of kind of ragtag band of bird species, which you see here, some of which are native, some of which are introduced, and those are going to be the focus for the rest of the talk. So the main thing I'm talking about today, as I mentioned, it's kind of a synthesis of what we're doing on Guam, and it's just in general determining the ongoing impacts of the tree snake on Guam's avifauna. There hasn't been a ton of research going on in Guam's birds, except for the Swiftlet research that's been going on for the last decade or two. But besides that, there's not really much known about Guam's birds, so that was what we were endeavoring to figure out. And the rest of this talk, I'm going to focus on a sort of generalized paradigm looking at various levels of biological organization. So starting at the individual level all the way up to the ecosystem level. And most of the talk is going to focus on the Sali or Micronesian starling. So at the individual level, I'm going to talk about patterns of age-specific survival. I'm going to talk about how those patterns cascaded to affect the Sali population size and age structure. Then we're going to zoom out a little bit, and we're going to look at sort of community level impacts of the tree snake on birds on Guam, specifically roosting behavior and roost site selection. And then finally, we're going to go back to the Sali to look at the impacts of the snake on a critical ecosystem service, that being seed dispersal. So I would also like to mention that the community and ecosystem levels are going to be through the lens of the landscape of fear. So this is a popular idea in the literature that basically the distribution and abundance of predators and of predation risk to the prey species will impact their behavior and kind of how they respond to these predators. So starting out with survival, we did a lot of radio telemetry the past couple years on Guam, and we also did a lot of color banding, where we put these little bands on Sali's legs, and you can identify individuals that way. And we use this information to essentially estimate survival across 3 main age classes. We also have information for nestling survival, which I'm not going to talk about today. That was kind of spearheaded by our collaborator, Julie Savage. But today I'm going to be focusing on the fledgling, juvenile, and adult stages, which you can distinguish here based on their sort of plumage differences. So fledglings are young birds that have just sort of fledged the nest and are still dependent on their parents. And the fledgling period in Sali lasts for about 3 weeks. And what we found here is, if you look on the left, this graph is a histogram of sort of fledgling survival across a bunch of different bird species and populations from a recent review paper. And if you focus on the black arrows, that's what we found for Sali fledgling survival on Guam in 2017 and 2018. So the key takeaway here is that Sali has incredibly low fledgling survival rates. In fact, if you look at 2018, you can see that they have the lowest survival rate ever recorded in the literature by far, only 12% in 2018. So the snakes are clear, and a lot of this is due to snake mortality. So the snakes are clearly having a large impact on fledgling survival. Interestingly, one other thing that we found, if you look at the juvenile phase, which are birds that have fledged from the natal territory and are independent but are not yet capable of breeding, so they're kind of just floating around the landscape, living their day, free-living birds, but not breeding birds. We found extended mortality into the juvenile period. So what you see here in this graph, this is from a recent paper we just published in the Condor, is you see probability of mortality on the y-axis, and those are the black lines. And then on the opposite side, you see the sort of the inverse of that, which is survival probability, which are the gray lines. And then the length of the post-fledgling period is indicated by that vertical dotted line. So about 3 weeks is when the birds become independent and leave natal territory. And what you see here is that for most sources of mortality, such as cats or the elements like weather, exposure, et cetera, you see that most of those level out around day 9 and are basically at about 0 from then on. So they quickly drop and birds are not dying from those mortality sources. However, if you look at the brown tree snake, what you see is that there is an elevated risk of mortality that extends all the way past the post-fledgling period into this juvenile period. And what this means is that survival steadily declines throughout this period. And what we found for juveniles is that overall survival, we found overall survival was about 35%, which is incredibly low. And this includes birds that are up to 109 days of age. So these are birds that you develop relationships with. So you're checking on them day to day or week to week, and then all of a sudden you find them end up in the belly of a brown tree snake. So pretty bleak. And again, this is quite low compared to other populations as well, similar to fledgling survival. When you combine these together and take sort of the product of fledgling and juvenile survival, you get a first-year survival rate of only 8%. So only 8% of individuals that fledge the nest are living to their first year, basically. So again, this is abysmal, and it shows just the incredible impact that the snakes primarily are still having on this population on Guam. However, one of the interesting things that we found is that adult survival is quite high. So adults are these are the Sali that you're probably familiar with seeing if you've ever spent any time on Anderson Air Force Base, the glossy black adults with the yellow eyes, and these guys have 87% survival. So in both years that we sampled, it was quite high. And so what's clear is that adults are more resistant to predation, although the mechanism is unknown. And this is something that we are currently working on, and that Haldra and Malia and some other people are collaborating on to try to understand exactly how these adults are living so much, are surviving so much better than these younger age classes. But this is likely one of the main reasons why this native species has been able to persist where so many others have perished on the island. So what are the population level impacts? Well, to address this question and address how the survival cascaded up to affect population level sort of characteristics, again, focusing on the Sali, we conducted a really intensive population census in 2018 on the population, which is centered, and most of the population resides on Anderson Air Force Base in the north. So this map was provided by my friend Shane Sires, and what you see here is we sort of gridded out the urban area on Anderson Air Force Base into these different sort of quadrants. We excluded the flight line due to access issues, but we believe that we were able to get a really good accurate estimate of the population. The reason why, which I'll touch on later, is because when these birds come into roost, they spend a lot of time in the forest in the afternoon, in the day, but in the afternoon they all kind of emigrate out of the forest back into the urban area, presumably because urban areas serve as sort of refugia to predation, because snake abundance is lower there and they're less likely to get eaten there. So because of this sort of weird quirk of this particular species, we're able to get what we believe are quite accurate estimates of the population. I'm not going to go into the details, but very briefly, we used resites of color bands, which I mentioned we were putting on all the birds that we were catching or taking out of boxes, nest boxes, and we used that to sort of estimate the overall population size. This is standardized methods. What we found was that the population is probably between 1,000 to 2,000 individuals. So that may seem pretty small, but if you take a sort of historical perspective, we've actually seen a 10-fold increase in population size since the last survey of Sali, which was done by Gary Wiles in the 1990s. He found that there were only 50 to 100 individuals left in the population. So this population was on the brink of being extirpated from Guam. And somehow they managed to come back, probably due to snake control methods and other control methods. But this is actually really encouraging in our mind. However, we are still seeing the effects of this on the population age structure. So if you look at the data from our population census, we found that 90% of the individuals that we were seeing were adults, and only 10% were fledglings or juveniles. And to give you some more historical perspective on that, the last survey of Sali, the survey that was done in the 1980s prior to Gary Wiles' survey, found that half the population was juvenile. So clearly, the snakes are eating a lot of these young birds. There's not much recruitment into the population, and they're having effects that are cascading up to impact the demographic structure of the species. Moving on to community level impacts, I mentioned I was going to talk a little about roosting. And so to do this project, we focused on sort of the remaining birds that are left on Andersen Air Force Base, these 6 species that you see here. And we're interested in roosting clearly because the brown tree snake is a nocturnal species. And so they're eating birds at night. That's kind of when it's most important what the bird's behavior is to understand how they're avoiding or getting eaten by these snakes. And so to do this, we did a really intensive survey again of the urban area on Andersen Air Force Base at night. And we actually replicated the survey twice. But basically we sampled over 800 trees, again, excluding the flight line for access reasons. And what you see here on the left are all the trees, little black dots that we sampled. And if you look here on the right, we found some really interesting patterns in terms of the sort of social aspect of this. So we found that a lot of these birds were aggregating in these large communal roosts. We actually found 56 communal roosts overall. And a communal roost we defined as any roost that had more than 5 individuals and or more than 2 species present in that roost. So some of these roosts had 5 or 6 species, up to 150 individuals in them, and just more in general, we found that almost a third of the trees in the urban landscape had birds roosting in them. So clearly, this urban area is serving as sort of a refugium from snake predation, and you can see here on the right panel that a lot of these communal roosts, which are the ones that are sort of shaded in different shades of red, are kind of concentrated near the center of base. And if you just pick random trees and compare to those random control trees, you can see that there are clear differences in the spatial distribution with all these roosts clustered kind of near the center of the base, presumably the areas where snake predation risk is lowest. We also found really cool spatial responses. So in this graph, you're seeing the kind of landscape classification in the left panel, panel A. And in the other 6 panels, you see the 6 bird species. And what you see here, what I want you to take away from this is that you see similar spatial patterns across these species. Again, you see that a lot of these species are kind of roosting more in the center of the base. And there's one exception to this, that is the Philippine collared dove. And you can see this here in the bottom right. And what I think is happening here is that these birds are kind of above the size threshold of the brown tree snake for predation. So I think they're too big to be eaten, at least when they're adults. And so I think that allows them to be more resistant to predation and have the sort of broader spatial extent of roosting. And you'll also notice that they don't have many communal roosts. So these birds usually roost alone. So again, they're not roosting socially, or they're not exhibiting the same spatial patterns as other species. And if you take it a step further, we actually found that body size is a really important predictor of the sort of use of communal roosts. So sociality here is just the propensity for communal roosting. How many of the roosts these birds use are communal roosts? And what you see here is there's a very strong relationship between body size, negative relationship between body size and communal roosting, whereby smaller species such as the Eurasian tree sparrow are essentially almost exclusively using communal roosts. About 80% of their roosts are communal. And then you have species like the dove, where only about 15 to 20% of them are found in communal roosts. So again, body size seems to be an important predictor of predation risk, which is then having this impact on sort of their behavior and how they're distributing themselves around the landscape. Moving on to ecosystem level impacts, we're going to again focus on seed dispersal here, which is a really important ecosystem service. And to address this issue, we again used radio telemetry of juveniles and adults, and we estimated home range size. And then we used a simple model developed by my friend and collaborator, Evan Ream here, where we basically took information that we knew on gut passage data, so how long it takes a seed to pass through the digestive tract of a solly, and then also the movement data and home range data to estimate rates of seed deposition in urban and forest habitats. And what we found, first of all, is that juveniles have larger home ranges, actually an order of magnitude larger. So this is just an exemplar, but we found this across the board for all the birds that we radio tagged. Juveniles have an average of about 370 hectare home range size versus only about 24 hectares for adults. And the other thing that you'll see here, which I'll talk about more in a second, is that juveniles seem to be using the forest a lot more than adults. And the reason why this is because adult habitat use is constrained by nest location. So this is an example of this adult, which nest is kind of at the near the edge of the urban area, and you can see that most of their territory is centered in the urban area, and they kind of make these occasional forays out into the forest, but they're not spending a lot of time out in the forest. And the reason why is because, as we suspect that due to this landscape of fear, because the snakes are more abundant in the forest, and also these birds kind of want to stay near their nest where their young are, that they're constrained to use urban areas more. And you'll see why that's important in a minute. We also found that there are really stark differences in habitat use between adults and juveniles. So on the left here, you can see that adults spend pretty much all of their time in the urban area, regardless of time of day. Whereas on the right, you can see that juveniles spend their day, and the first thing in the morning, they leave the urban area where they're roosting, they head out in the forest, and they spend most of the day foraging out there. And then again, as I mentioned in the population census, most of these birds end up coming back in the afternoons, which is why you kind of see this increase in urban habitat use by juveniles throughout the day. As I mentioned, we use this model to try to understand how this impacted seed dispersal. And so basically, we would do telemetry sessions of about 2 hours, 130 minutes. And in those 130-minute period, we essentially had, we know that the solly gut passage time on average is about 40 minutes. So basically, that comprises about 9 potential dispersal events if you move a 40-minute sliding window across the 2-hour telemetry session. And then we did this simple model where we said if a solly was in the forest at the first time point and then 40 minutes later, it was still in the forest, we consider that a potential forest dispersal event. Whereas if it started in the forest and then 40 minutes later, it was in the urban area, that was a potential urban dispersal event. So again, this is not necessarily a realistic depiction of where they're depositing seeds, but it's sort of just a relative estimate of their impacts.
Henry, you have 1 minute left.
Okay, basically what you see here, as I mentioned earlier, is that adults are spending most of their time in the urban area, whereas juveniles are spending most of their time in the forest. And if you look at the last column, you can see that juveniles are basically doing most of the forest dispersal, whereas adults are not dispersing anything in the forest. So based on this landscape of fear, you see this difference in behavior across these age classes, which is having huge impacts on seed dispersal. So in summary, I think I've shown you that first-year survival is really low. Adult survival is probably why the species is so persisting on the island. And this is having impacts in the population with 90% adults in the population. I've also shown you social and spatial responses to the landscape of fear, a lot of which is impacted by body mass of the species. And we also have shown that this cascades up to impact seed dispersal and is having important effects on where and how these birds are dispersing seeds. So with that, sorry for going over. Thanks for listening, and I probably don't have any time for questions, but hopefully you guys enjoy the talk. Thanks a lot.
Thank you, Henry. People can email him if you have any questions. I can give you his email. And we're going to move on to our next talk. Nathan Johnson is going to tell us about the population ecology of the Marianas swiftlet on Oahu. Thanks, Henry.
See you later.
Yeah. Chalk talk? The chalk talk? I said, do you want a chalk talk? I have a clicker. I just need to set it up. Yeah. I have a clicker. I just forgot to set it up because that was a Zoom one. So you are the guinea pig, yeah. Where is he? I just figured to unplug the power and not the.
Sorry. Can Can everybody hear me okay? Oh, actually. I don't need this. Does this come out? Because I don't want to. Can I take it out? Just pull out. All right. Good morning, everybody. Everybody hear me okay? So I'm going to talk a little bit about my master's research on Oahu with the Marianas swiftlet. We had the unique opportunity to study the population ecology of Marianas swiftlets over 5 complete annual cycles between 2006 and 2011. This was largely in part due to Steve Mosier, who was living on Oahu at the time and spent his personal time monitoring nests that I had set up in 2005 and 2006. So with that, I hope you guys learned something interesting about these unique birds. So Mariana swiftlet measures 10 centimeters, weighs about 6 to 8 grams. They're endemic to the Mariana Islands. Been federally endangered since 1984. They occupy limestone caves where all the roosting and nesting activities occur. And they're unique in that they use echolocation to navigate within cave darkness. Mariana swiftlets were formerly distributed on 5 islands in the Marianas. They currently persist on 3, Saipan, Agiguan, and Guam. Swiftlet sightings on Tinian occurred 1945 and 1976, were believed to be transient birds from Saipan or Goat Island. They did find bones in 94 of swiftlets in a cave in a rock shelter, but there was never any records of breeding of swiftlets on Tinian. Swiftlets were known to nest in at least 6 caves on Rota. The last sightings there occurred in 1976. So for those translocation fans, swiftlets were captured on Guam and released on Oahu in 1962 and 1965. This was done for aesthetic purposes and apparently to augment insect control. Believe it or not, these were sponsored by the state of Hawaii and a society called the Hui Manu Society, who specialized in the introduction of exotic bird species in Hawaii. This is the 60s. So in 1962, 175 birds were captured on Guam and moved in wooden crates on a commercial airliner to Honolulu. And 1 bird died in transit. 1 egg was actually laid in transit. So those 174 birds were released down in Niu Valley in the south of Oahu. 3 years later, to make sure that the population establishes itself, 210 more birds were captured on Guam, and all were successfully released up in Waimea Valley in '65. So swiftlet sightings started to cluster in 1969 in North Alava Valley, which is where in 1978 the swiftlet was first documented breeding in a tunnel in the Ko'olau Mountains. This tunnel is a single entrance straight passage about 55 meters long. It averaged about 1.7 by 1.3 meters. It's wet and muddy throughout the year. And we conducted evening arrival surveys with night vision devices from inside the cave. So the latest estimate that I have were 142 volant swiftlets that were occupying the tunnel at the time. So I was interested in nesting biology for one aspect of my thesis. We described and measured nests and eggs of the swiftlet on Oahu. We characterized the nest sites, and we calculated incubation, nestling, and nesting periods. So swiftlet nests on Oahu were circular cups of liverworts, mosses, and ferns. They were adhered to the tunnel walls with mucus-like salivary cement, which you can see in the photo there. Mean construction period from the point when we first observed salivary cement on the tunnel walls to the point where an egg was in the cup was about 34 days. Outer length and outer width was about 6 by 6 centimeters. All active nests were built in total darkness in the rear 22 meters of the tunnel. Swiftlets laid one white egg that was elliptical and non-glossy on Oahu. Mean length and breadth were about 19 by 12 millimeters. Mean incubation period of swiftlets on Oahu was about 24 days. Mean nestling period about 55 days for a total nesting period of 79 days. So we monitored active nests from January 2006 to January 2011. Active nest sites were identified with uniquely numbered reflective tape tags. Nest checks were on average about a week apart. We verified nest status, so whether there was an egg nestling, whether it fledged, failed, or it was empty, with night vision goggles, a headlamp with a red filter. Since the white light freaks them out, the red filter didn't freak the swiftlets out, or a video camera with infrared that was later reviewed. Because this is such a tight tunnel, nest detectability was 100%. So for each year and all years, I calculated hatching success as the proportion of eggs that hatched, fledging success as the proportion of hatched nestlings that fledged, and nest success as the proportion of eggs that resulted in a fledged nestling. So early on in this study, I documented rat predation in the tunnel. 18 swiftlets were dead. So we kicked in rat trapping in March 2006 and continued it until January 2011 at 43 sites. 38 of the sites were outside the tunnel and 5 were just inside the tunnel, but not too far to where we might attract rats to the colony. And the annual mean number of traps that were active and baited per night increased throughout the study period. Total of 406 rats were captured. 18 of those were inside the cave and 388 out. So this is swiftlet nesting activity over 5 annual cycles on Oahu. Notice the cyclical breeding pattern that continued throughout the year. Active nests were observed in every month of the year. Rat predation events occurred in 2006, 2007, and 2009. The 2006 event essentially interrupted that breeding season when 18 adult swiftlets were found dead. 2007 predation event essentially delayed the onset of that breeding season when 7 adult swiftlets were killed. And in 2009, that essentially terminated the 2009 breeding season when 10 adults were found dead and 17 nests failed. So in all, there were 35 adults that were killed and 14 nestlings during the study period. So it's important to recognize here that swiftlets completely recycled their nesting activities after these rat predation events. And also, interestingly, the swiftlets did not abandon the tunnel after these predation events. So 478 eggs were laid throughout the 5-year period. 86% fledging success and 63% nest success rank among the highest of any aerodroma swiftlet that's known to lay one egg. Reproductive output and success in 2010 was probably tied to the highest number of rat traps that were deployed and the highest number of rats that were captured that year. Low nest success in 2006 and 2009 could probably be attributed to rat predation events. So reproductive activity and success probably wouldn't have been so high without that rat trapping. So with nest failures, the primary source was we found a lot of eggs in the water or mud. And, 5 minutes, okay. Thank you. So we found a lot of eggs in the water or mud. Reasons for this are unknown, and I can talk offline with folks about this, or I can answer questions about it. I had a slide about this, but that was the primary reason. The eggs were basically out of the nest in the mud or the water. One of the other failures, the nestlings that were found in the water or the mud, just reading the literature, swiftlet nestlings are very hypersensitive in the nest. So they tend to crawl around a lot, and so it's possible that these guys may have, just lost their balance and fell out. They also, swiftlet nestlings also back themselves up over the front rim of the nest or the side and defecate. So that's also another possible reason that they just lost grip and fell out. Rat predation covered. Nests falling off the tunnel wall. Heavy rains would cause a lot of water to drip down the walls in the tunnel. And so that water probably saturated these nests to where the weight of the nest was too much to where the salivary cement couldn't hold them up any longer. General conclusions. Marianas swiftlets on Oahu breed throughout the year. Eggs found in the water or mud was the primary reason for nest failure. Rats are a predator of swiftlets on Oahu, and it's important to recognize their effect on adult survivorship and nest success. And limited tunnels and rat predation may preclude population growth on Oahu. This study was among few that have examined the full life cycle biology of an aerodroma swiftlet, let alone over 5 years. So having a population on Oahu to learn more about natural history and possibly test reintroduction techniques is an important advantage for swiftlet conservation efforts. Finally, results of this multi-year study provide essential life history information for swiftlet recovery and reintroductions in the Marianas. I'd like to thank many folks at Oregon State University, in addition to USGS FRESC and U.S. Fish and Wildlife Service. With that, I'll take any questions. I guess I'll keep this. Do I have a few minutes, or? 3 minutes for questions? Oh, sure.
Okay, I'm afraid to talk now. So you have a cyclic nestings or peak in the summertime and less nesting. Did the swiftlets come back and roost in their nests, even when they weren't actively nesting?
No, they're roosting on the walls. Is there a mic? Sure. Well, overall, obviously, it didn't affect them. I mean, I'd have to tease that data out, the individual nest sites. But the big picture graph after those predation events, they certainly came back. And that could be an artifact of just limited tunnels on the island. Now, there are hundreds and hundreds of these tunnels on Oahu that were basically tapped for irrigation back in the day for sugarcane fields and homesteads. And this particular one was, I think, had enough dryness in the back where they could adhere those nests. I checked a lot of tunnels and they were just soaking wet and too wet for swiftlets. So, but the tunnels could be limiting there. But it's amazing that they found the place in the first, since the 60s. Is that it? Okay.
Thank you. Our next speaker is Marcos Gorson. Oh, sorry. Our next speaker is Marcos Gorson, who will be continuing to talk about Mariana swiftlets, this time on Guam, with nests and population counts with thermal videography.
Thanks very much for attending. Can you hear me back there? No? Hello, hello. Thanks very much. This is a collaborative project with the University of Hawaii at Hilo and the USGS and NAFAC Marianas. I'm going to present some preliminary results from work we did in April and just last week on Guam. As we know, the Mariana swiftlet is limited in population distribution to the Navy base, Magazine in southern Guam. Nathan Johnson's estimate in 2016 puts the birds at about 1,500 total population, and most of it is in Malak Cave. There's about 80% of the population in that cave. The species is a roosting and nesting cave obligate that lays a single egg in its nest, as Nathan showed us. And it adheres the nests on the ceilings and walls of the cave, often out of reach of the brown tree snake. There's been a long series of population estimates since the 80s, and you can see from this graph that it varies considerably over time. I mean, there's some good news in there in terms of the upward trend, but there's a lot of year-to-year variability. Some of that's seasonal, some of that's year-to-year, and some of that is probably sampling error. These birds can be very difficult to count during the exit and entry counts. And this causes problems with assessing management efficacy and also the overall trend of population. So let me talk a little bit about some new thermal imaging methods that we applied to develop perhaps an improved estimate of getting that population size, and an alternative metric that might also work for assessing management effectiveness. The techniques that we've used were originally developed for tracking bats at wind turbines at night, and very bats that are distant targets. And many of these were rare events where you'd collect thousands of hours of video and process them using software. The work that we've done has relied extensively on these workhorses here, these Axis Communication thermal cameras. They have a fairly high pixel resolution, and they can record to SD card or an external drive for later analysis. We worked at the 3 caves. I'm shown here the Fachi has 2 entrances, west and east. And this is Maymong, the entrance to Maymong, and Malak Cave. I'm going to show a series of slides that are focused on the cluster of birds in the area encircled in yellow. So we tested a number of camera configurations on swiftlet caves last April and last week. And this picture shows the entirety of the colony on the ceiling of Malak Cave. And And the blue circles here show the actual clusters themselves. So the next series of slides is going to focus on that particular area. Let's see if I can get this to play. So this is that cluster, and this is a recording at nighttime. So you have all the adults at the roost. And we see considerable amount of activity of birds throughout the night at the roost. There's a lot of what look like young fledglings attempting their first flights right before they venture out. And a lot of the adults are situated on their nests. So again, what we see is we can often make out the pairs through longer view of the video footage. We record for up to 48 hours at a time, and we analyze this both at day and night time. And you can see single birds roosting on the cave walls, juveniles in flight. And during the daytime, what we see is the majority of adults are out foraging, so there's quite a bit fewer birds, and what remains are adults incubating nests and the nestlings that are visible in the nest. And from this, we can start identifying the number of birds in each of these clusters. So for this particular video frame, we counted 54 nests. So the next series of slides is going to be same general area, but with a different camera lens. And what we see here is a high-speed replay throughout the night. It's like an 8-hour clip running all the way to dawn. And we use this kind of a long-term monitoring to look at the points of light, the thermal blobs that are consistent over both day and night. And from this, we can start mapping out nest locations and distinguishing them from just roosting birds, which I kind of wink in and out of the image here. So the square showed the last area I've shown, and I'm going to kind of talk a little bit about these other clusters. And again, by comparing these over a series of many frames, we can start to map these out and look for consistency in their location. And this is all kind of manually done. Takes a day or so. And you can start mapping these out. You can see a roosting bird that kind of shows up, disappears. And again, we get a very accurate estimate of the number of nests in this particular sample period. And we did this for the entirety of the colony, and for last April got a total of 284 nests that were either with adults on eggs or nestlings and juveniles still in it. We did this again for a Maimong cave using a kind of a lower resolution camera, and we got a count of 68. And we did this for Fachi with an even lower resolution camera and got 8 nests that were consistently there throughout the 24-hour period. So to summarize, the thermal video system we're using provides high-resolution recordings that can be manually analyzed for behavior and bird and nest counts. Another camera technique that we're exploring is the use of near-infrared, again, to look at behavior and nest attendance, and it's also going to be analyzed to give us an index or a ratio of the number of adults seen at nest during the daytime when you can count them and there's fewer birds, and the nighttime when you have everybody back in the nest or at roost. And this can be used as a multiplier to get a population estimate. So we're currently developing computer vision methods for automatically tracking and counting birds, as well as bats. Again, my interest is originally with bats, and this has a lot of applications for bat studies. And The applications will include automating this nest count process that's currently done by eye, and the tracking of bird movement to and from nests to determine nest attendance rates, and the counting also of birds at the cave entrances, both in their exits and entries, to get at a population estimate, much as has been done in past surveys, typically by eye. We feel confident that this method is going to provide us a very good series of metrics to assess management efficacy, especially brown tree snake control. We're also exploring the use of time-lapse cameras to get at this nest attendance and nest use over long periods, perhaps as long as months. Okay, and that's what we got. I'll take any questions. Yep. Okay, thanks very much.